Hello and welcome to the session. In this session, we are going to talk about several different types of separation techniques, and we are going to focus more about the process of centrifugation. This information is coming from this book chapter, Centrifugation and Separation Techniques, from this book, Understanding Bioanalytical Chemistry. So first thing I'm going to talk about is why we need to separate, why we need to purify substances. One important reason for separation of substances, and in this particular context about different types of biomolecules, is that in order to study those molecules, those biomolecules, they need to be purified and separated to a certain extent so that we do not have any interaction from other types of molecules. So for one reason, in order to study, in order to know about the structure, in order to know about their functions and properties, the biomolecules, they need to be separated and purified. Another important reason why this process of separation is important is that many biomolecules, they are used as therapeutic agents, or they are used for several other applications like food additives, supplements, and several other applications. So in order to act as an ingredient suitable for human administration, so these molecules or substances, they should be purified or separated to an acceptable level. So for these reasons, that is in order to study the properties and to analyze and evaluate, and also to use these as supplements, therapeutic agents, or additives, these molecules or these substances, they should be purified and separated to a sufficient level. Here in this topic, we are going to talk about several different types of separation techniques, and the primary focus here is going to be pertaining to centrifugation as a separation technique. So among these methods, the first thing is about dialysis. Dialysis is a separation technique which is based upon the difference in the concentration of substances. Dialysis is carried out by using a dialysis membrane. As you can see here in this figure, this is a container, and inside the container we have a suitable buffer for dialysis, and this is a dialysis tubing. The dialysis tube has pores in it. Now this is a magnified view of the dialysis tube. So these are the molecules which are present on the inside of this tube, and this is the buffer present on the exterior. As you can see here, that there are small pores which are present in this dialysis membrane. Now the molecules which are abundantly present inside, that is their concentration is higher inside the tube and their concentration is low on the outside of the tube, they can move down their concentration gradient. That means they are going to diffuse out through this dialysis membrane. Since in this example you can see these small white circles, they are small molecules and their size is less than the pore size of the membrane. So these molecules are going to diffuse out into the buffer. On the other hand, the other type of molecules which are dark gray circles, so these are larger molecules and hence they are unable to pass through this membrane. So in this manner, the small molecules, they are going to diffuse down their concentration gradient, and therefore they could be removed from this solution. One of the important applications of dialysis in biology is to carry out the removal of salts. For various applications, we need to add salt to the solutions of proteins or other different types of biomolecules. Later, in order to get rid of that excess of salt which was previously added, we can carry out dialysis. One such application regarding proteins is where we want to precipitate proteins. So proteins could be precipitated by addition of salt, for example, ammonium sulfate or some other suitable salt. Once the required protein is precipitated and dissolved in the solution, for many applications we need to get rid of this salt. So this salt could be removed from the protein solution by carrying out the process of dialysis. This process to remove salt from a given solution is called as desalting, that is, to remove salt. Dialysis tubes are available with different cutoff sizes. The term cutoff means that what type of molecules can pass through the pores of this membrane. So for example, if the cutoff limit for a dialysis tube is 5 kilodalton, that means molecules with size less than 5 kilodalton can move through this. Molecules having size larger than 5 kilodalton are unable to pass through this membrane. So in this manner, we can use dialysis membrane with various cutoff limits and can use it for carrying out dialysis. Say for example, proteins are large molecules and the salt which is added is of course a small molecule. So we can use a suitable cutoff range of the dialysis membrane, and eventually that would result in separation of the salt from the protein solution. After some time, when the salt is going to be removed from the protein solution and it is going to diffuse out, after some time an equilibrium of salt concentration will be established inside and outside of the dialysis membrane. So therefore, the buffer which is present on the outside has to be periodically replaced, so that at that equilibrium point, otherwise no salt will be removed further. So therefore, the buffer has to be removed and a fresh supply of buffer has to be added after some interval of time. So this change of buffer again establishes a concentration gradient, and now the salt will be moving down the gradient. That means from the inside of the tube to the outside. Dialysis could also be used for concentration of a given solution. For example, let us say if we have a protein solution. So if the protein solution is placed in a hypertonic solution, the buffer outside is a hypertonic solution. So the protein solution becomes hypertonic. A hypertonic solution has more free water in it. Hypertonic solution has less free water in it. So if you place a solution in a dialysis tube and place it in a buffer which is hypertonic, so free water molecules will start to move from the inside of the tube to the outside. So in this manner, the volume inside the dialysis tube will become less and the substance is going to be concentrated. This is another important application for the process of dialysis. Dialysis can not only be used for separating salt from a given solution of biomolecules, but it could also be used for separating small molecules from larger molecules. By using a suitable membrane of a suitable cutoff limit, we can carry out separation on the basis of mass. The process of dialysis is relatively slow and it is dependent upon the diffusion of substances through the membrane. The process can become rapid by application of electrical current. If we apply potential difference in this buffer, the molecules which are to be separated, if they're charged, they are going to migrate rapidly towards the cathode or the anode, depending upon the nature of charge on them. So electrodialysis is a mechanism of dialysis in which we apply potential difference and apply electrical current. As a result of that electrical current, the charged substances which are permeable, they are going to pass through the membrane rapidly and they are going towards the corresponding electrode. In this manner, the process of dialysis could be carried out rapidly. Some people with renal dysfunction or renal disorders, they also undergo dialysis. That is called as hemodialysis. The term hemodialysis refers to dialysis of blood. So the kidneys, they remove the waste materials from blood, and in those people who are suffering from kidney dysfunction, they are unable to remove the waste materials properly. So during hemodialysis, the blood from a patient is allowed to exchange the waste materials through a selectively permeable membrane with the dialysis fluid. So in this manner, the waste materials are removed from the blood of a person and they are diffused away into that dialysis fluid. So in this manner, the waste materials from blood could be removed. This process is called as hemodialysis. The next type of separation technique is called as precipitation. Precipitation is based upon solubility. Substances are soluble in a given solvent if they are able to interact with that solvent. For example, salt dissolves in water because salt is able to interact with the solvent and get solvated. So the substances which are insoluble in water, they are insoluble because they are unable to interact with the solvent. So any solute will remain soluble, will remain in solution if it is able to interact with the solvent. If through some mechanism we can decrease the solubility, or in other words, we decrease the interaction between solute and solvent, then the substance or the solute is going to precipitate. It will become insoluble and it is going to precipitate. So if through any mechanism we decrease the interaction between solute and solvent, so in that way the solute could be precipitated. So this process of changing the solubility of a substance and taking it out of the solution is called as precipitation. Precipitation could be done in several different manners. For example, one of the ways to precipitate the biomolecules is by using less polar solvents. Water is more polar and hence it can dissolve polar substances in it. If we decrease the polarity of the solvent, then the solubility of solutes also decrease. For example, one method to precipitate substances is to add alcohols. Ethanol, methanol, these could be used for precipitation of different types of biomolecules. For example, nucleic acids including DNA and RNA, they are precipitated by using ethanol, sometimes by using isopropyl alcohol as well. Alcohols, although they are polar, but their polarity is less as compared to the polarity of water. So when we add alcohols to an aqueous solution, now the solvent contains less polar molecules in it. Therefore, the interaction between solute and solvent decrease, and that is going to result in precipitation of the solutes. Another important agent which is used for precipitation is ammonium sulfate. Ammonium sulfate is widely used for the precipitation of proteins. When ammonium sulfate is added up to a certain point, it is going to compete with the proteins for solvation. When ammonium sulfate is added in excess, now ammonium sulfate is a salt. When dissolved in water, it is going to ionize into its constituent ions. And these ions are charged, so they interact with water molecules. Now, ammonium sulfate, the salt ions, they are small in size and therefore they can interact with many water molecules. So the salt ions, they are going to compete with proteins for solvation. Proteins are large molecules and they need many water molecules to be solvated. Salt, on the other hand, is a small molecule and could be easily solvated by less number of water molecules. So the free water molecules, they will be engaged in solvating the salt, and only a little water molecules will be available to solvate the proteins. So in this way, interaction between proteins and water molecules will decrease, and hence this would result in decreasing the solubility of proteins. Other substances like acetone, trichloroacetic acid, they could also be used for precipitation of proteins. And lastly, antibodies could also be added to precipitate proteins. Antibodies are proteins which are produced by the B lymphocytes. Antibodies are specific to bind to a particular type of an antigen. So if we have antibodies which are specific to a given protein which is present in a solution, when antibodies they bind to a given protein, an antigen, then they produce a complex, they form an aggregate, and the solubility of this aggregate is low. In this manner, addition of antibodies to a solution of an antigen would result in decreasing the solubility of that antigen. In this way, antibodies form a complex, an aggregate with antigen, and this complex precipitates. So in this manner, a required protein could be selectively precipitated from a solution. So sedimentation is a process in which substances are going to settle down in a given medium. Centrifugation is based upon the process of sedimenting substances by applying centrifugal force. Previous expression that the force applied is dependent upon the radius of the rotor as well. So therefore, if we carry out centrifugation at a given speed, say 4000 rpm in one type of a rotor, then the force applied would be different if the rotor is changed. So if I carry out centrifugation in a rotor of 20 centimeter radius at 4000 rpm, then the force applied would be different if I change the rotor of a different radius and I spin it at 4000 rpm, then the force applied would be different in this case. So therefore we have this expression to describe rather than in terms of rpm, we can describe the relative centrifugal force applied, and which is given by this expression shown here. The relative centrifugal force is actually going to compare the force exerted onto the specimen with reference to the acceleration due to gravity. It is also expressed in terms of g's. So that means if an object of a given mass is going to accelerate under the influence of gravity, then there will be a certain amount of force applied on it. So the relative force applied is going to be in relation to the force applied onto that object when it is going to move under the influence of gravity. So this is called as the relative force, and it is expressed with respect to the acceleration due to gravity, expressed in terms of g's. So here it shows the basic construction of a centrifuge. A centrifuge contains a motor, and there is a rotor which is attached to that motor. So the motor rotates and it is going to result in spinning of the rotor. Inside the rotor there are slots where we can place the centrifuge tubes. And when the rotor spins, force is applied onto the specimen which is present in the centrifuge tubes, and therefore those substances, those molecules present in the tube, they could be separated from one another. There are different types of rotors available. So in one type of rotors, as shown on the right side, these are called as the fixed angle rotor. The tubes are placed in the rotor here at a certain angle, and these tubes are present at a fixed angle in the rotor. So therefore such rotors are called as fixed angle rotors. The ones shown below, these are called as the vertical rotors because the tubes are placed in these rotors in a vertical position. And the third one is called as a swinging bucket rotor. These rotors have a hinge, have an attachment here at the top of the tube. So when remainder of the tube is free. So when this tube is at rest, it is going to be in a vertical position like this, but when force is applied, the free end of the tube is going to project outward. So now the tubes become horizontal during spinning. So that is why it is called as a swinging bucket rotor, because the tubes are swinging in this rotor. Now, in a fixed angle rotor, the direction of force applied is outside like this. So therefore, the substances are going to sediment on this outer edge of the tube. In a fixed angle rotor, the direction of force is applied outward, so therefore the substances are going to sediment on this entire surface of the tube facing outside. In case of a swinging bucket rotor, so the force is applied in this direction, outward direction, so therefore the substances are going to migrate all the way through this tube, and they are going to be sedimented at the bottom of the tube. So the position at which sediment is going to form is going to be different in all three different types of centrifuge rotors. For various applications, we use different types of centrifuge rotors. Centrifuges are classified based upon the speed at which they can spin. Generally, centrifuges having speeds of about 6,000 Gs are classified as low-speed centrifuges. Centrifuges with speeds reaching up to 50,000 Gs are called high-speed, and centrifuges with speeds 100,000 and above, they are called as ultra-centrifuges. Centrifugation could be done in several different ways. One of the type of centrifugation that is called as differential centrifugation. In this process, a specimen is centrifuged at a low speed first, so larger substances are going to sediment, and other substances, other molecules are going to remain soluble. So here, as you can see in this example, there is a specimen present in this centrifuge tube. So initially the tube is centrifuged at a low speed, in this example 600 Gs. So those substances which are larger in size or their solubility is less, so they are going to sediment at the bottom of the tube, and rest of the molecules are going to remain soluble. The substance which sediment is called as a pellet, and those substances which are still soluble, the liquid here is called as the supernatant. The supernatant here would be containing soluble substances in it. So we take out this supernatant by using a pipette and transfer it into a new tube. Now this is centrifuged at a relatively high speed, in this example 3000 Gs. After this, some more substances are going to sediment at the bottom of the tube, and other molecules will remain soluble in the supernatant. So then we collect the supernatant and subject it, transfer it to a new tube, and subject to centrifugation for at a higher speed. In this example, 15,000 Gs. Then some of the substances would again sediment at the base of the tube, and other soluble material will remain in the supernatant. So supernatant is then collected and then subjected to centrifugation at even higher speeds. In this example, 100,000 Gs. And therefore, the substance which is the material which is sedimented is collected here, and rest of the soluble substances remain in the supernatant. So here, in this way, we can carry out centrifugation in this differential manner, that some substances are separated at a given speed, they are going to sediment, other substances are going to remain soluble, and then we centrifuge at a higher speed, and some more type of substances, they could be separated from the specimen. In this way, we carry out a series of centrifugation steps, and with every step, the speed of centrifugation is going to increase. So this method is going to fractionate the substances and the molecules based upon their ability to be sedimented at a given speed. In this manner, the constituents of a given specimen could be separated from one another. This process is called as differential centrifugation. Typically, differential centrifugation has been used for the separation of cellular components, as shown in this example. The other type of centrifugation is called as density gradient centrifugation. As the name say, there is going to be a gradient of density in the centrifuge tube. There are two modes. One mode of density gradient centrifugation is called as zonal centrifugation. In this method, a density gradient is obtained by pouring the liquids inside the centrifuge tube. Different type of substances could be used to produce a density gradient. For example, sucrose density gradient is made by producing different concentrations of sucrose. In a density gradient, first we put the highest density solution at the base, then add a solution with relatively less density over it, then pour a third solution with less density, and then eventually we keep on layering these solutions of varying density. So the density at the base of the tube is going to be high, and the density towards the top of the tube is going to be low. Now, after producing this density gradient, the specimen is overlaid on top of this centrifuge tube. So that means when the specimen is placed here, the density is going to increase down the tube. Now we carry out centrifugation in a swinging bucket rotor. In a swinging bucket rotor, the tube is going to become horizontal, so therefore the substances are going to migrate from the top of the tube to the bottom. Now when the substances are going to migrate through the tube, they are going to experience an increase in the density. When the density of a given substance becomes equal to the density of the medium, that substance will stop migrating and is going to be localized in that region. Now another substance having a higher density will be able to pass through this region, but when the density of that substance become equal to the density of the medium, so it is going to stop migrating and will be localized in this region, in this zone of density. Substances with even higher density will be able to migrate further, and eventually they are going to stop migrating when their density becomes equal to the density of the medium. Those substances in the specimen which have a very high density, they would be able to migrate all the way through the tube, and they are going to sediment at the bottom of the tube. In this way, the molecules or the constituents of a specimen, they could be separated from each other into these visible zones, where each zone correspond to a different density, and the substances, they are going to localize to a certain density region if the density of those molecules become equal to the density of that zone. So in this manner, the substances or molecules could be separated from one another based upon their densities. Another mode of density gradient centrifugation is called as isopycnic centrifugation. Isopycnic centrifugation has an even or a homogeneous concentration of the species when centrifugation is going to take place. For example, cesium chloride is a salt, and it is widely used for separation of nucleic acids through density gradient centrifugation. So during isopycnic centrifugation, we have a mixture of the density medium and the specimen which is to be separated. So the specimen and the density medium, they are both are homogeneous in this tube, and now we subject this to centrifugation. As centrifugation is taking place, the density medium is going to partition and it is going to form forces applied in this direction downward. So a gradient of density is established during the course of centrifugation, and this is a continuous gradient. Now the molecules which are present in the specimen are going to be separated from one another as they migrate through this density gradient. So on top of the tube would be low density, and towards the bottom of the tube would be higher density. So substances are going to be separated from one another based upon their difference in their density. Later, once the process of density gradient centrifugation is completed, so these various layers containing different types of substances with varying densities, they could be collected by pipetting out these various layers of the density medium. Depending upon the nature and the application of a centrifuge, so there could be two important natures of the centrifuge. The analytical centrifuge, as the name say, is meant for carrying out analysis. Typically, analytical centrifuges, they have a small sample size, and they have a built-in optical system to analyze the substances or the molecules which are being separated during the run time as they are going to be separated. As opposed, the preparative centrifugation is meant for producing larger amount of the specimen. They can handle larger sample size, and generally they do not have optical readouts in them. So the fractions are collected on a large scale, and then the fractions could be analyzed later on. Now, a few important things regarding the practical considerations. One important thing is the scale at which centrifugation is to be done. Say, for example, if you are going to centrifuge at a scale of like 1 or 2 milliliters, then a small benchtop microfuge would be suitable. However, if you want to use a larger volume of the specimen, then a suitable size centrifuge tube with a suitable rotor should be used. Another important consideration is the size and the shape of substances which are going to be separated. So therefore, a suitable force should be applied to separate molecules of a given size from others. That suitable force is applied by using a suitable speed of centrifugation. Cooling is also needed in for several applications. Different types of biomolecules, they may be sensitive to temperature. For example, you may be carrying out centrifugation of a protein which is sensitive to temperature. So therefore, this should be done in a refrigerated centrifuge which has a cooling function in it. Also, centrifuges which are operating at very high speeds, so when the rotor is rotating at a very high speed, the frictional force of air results in increase in temperature. So therefore, those centrifuges which are operating at very high speed, they also have a built-in cooling function in them. Another very important consideration is to have a balance of mass across the rotor. As the rotor is spinning, we have seen this previously, that the force applied is proportional to the mass. If the tubes, they contain different mass of the specimen, then the force applied on different parts of the rotor is also going to be unequal. As a result of this unequal force, there will be stress on the motor and the shaft on which the rotor is rotating. And if it is imbalanced to a greater extent, then this may cause damage to the centrifuge as well. Many of the centrifuges, they have a built-in alarm, so that if the machine detects an imbalance, it is going to produce an alarm. It is going to sound an alarm, and hence you should take care of it. So it is important to balance the tubes before putting them in a centrifuge so that the distribution of mass across the rotor should be equal. Another important thing is the capacity of the tube. As a general rule, the tubes, the capacity of whatever the volume the tubes can hold, generally we do not fill them to their maximum capacity. So, depending upon the sample size, we can select the tubes of a suitable capacity, and then an appropriate rotor should be used for carrying out centrifugation. For several applications, the tubes and the rotor, they should be cleaned and sterilized. Say, for example, if you want to centrifuge a bacterial cell culture to separate the bacterial cells. So in this application, we need to have sterile tubes. So the centrifuge tubes from many different suppliers, they can be autoclaved. So through autoclaving, we can sterilize the tubes. All types of tubes cannot be autoclaved. Similarly, for several applications, the centrifuge tubes, they could also be sterilized by using ethanol. Rotors should be evaluated, analyzed before every spin, before every centrifugation process. You should take a quick look at the rotors to see is the rotor clean, is there any damage to the rotor or not. So you should always inspect the rotor before use, and once the process of centrifugation is completed, you should clean the rotor as well. Those centrifuges which have a cooling function, they also accumulate condensed water in them. So after use, the centrifuge should be maintained at room temperature, and the water which has condensed inside the centrifuge machine and the rotor, it should be cleaned properly. And when you are going to run a centrifuge, you are expected to stay close to the centrifuge, especially when the centrifuge is accelerating. Once it has reached the set speed and now the centrifuge is running smoothly, so yes, you can step aside for other applications, for other work as well. But as long as the centrifuge is accelerating and is going to reach the set speed, at least during that time, you should stay close to the centrifuge and observe is everything is working fine. If you hear alarms for imbalance, or if you hear any noise coming from the centrifuge, you should immediately stop the centrifuge and inspect it. If you are unable to inspect, then you should seek assistance about this.